Hey, hello guys, welcome, Manakam, Mandanam, Namaskaram. Today we are going to see Biotechnology Seminar 2. Already we have covered Seminar 1. If you haven't watched it, please go and watch it. Then only you will be able to understand everything perfectly. Okay. So now we are going to see tools. Okay. So tools of recombinant DNA technology. Great. In the beginning even we have started to do everything properly. Okay. So tools of recombinant technology, recombinant DNA technology, na? Okay. So what does it mean? See, to produce or to create rDNA or to create recombinant DNA, you will have to use some tools. Okay, you will have to use some tools. To produce recombinant DNA, you will have to use some tools which are part of technology. Okay, so what are those tools? That is what we are going to see now. Well, first, very foremost and very important thing is going to be your restriction enzymes. Okay, your restriction enzyme plays a very vital role in it. Okay, then after this we are having gel electrophoresis. Okay, we can say gel electrophoresis, that is actually used to see the DNA fragments. Well, not here. Okay, so we'll be mainly studying about this one. Then we can also say gel electrophoresis, that's what we have over here. Okay, gel electrophoresis. Okay, then what else do you want? Actually, this is not used to create rDNA, but this is used to see DNA molecules in a very good way. So we will just take it out. Okay, we will take it out. We will take this out. Okay, now we will come back to here even. You will have the need of desirable gene. Okay, desirable gene. Then you want a vector. Okay, then you want the particular place where you have to insert that desirable gene. Okay, so we can say it as, okay, so this one is also called as alien gene. Okay, this one is also called as alien gene. Okay, now then also you want that base, that plasmid. Okay, we can say it as actually vector is itself. Okay, in vector even you will have that plasmid. Then okay, so these are those very important things. Then also you have, okay, vector will also have that cell. Okay, everything we can say. So these are those very important things. And also you want genetic engineers, you want advanced technologies, you want microscopes, everything you want, but that at all we will not take. We will just take these three which are very important to create your rDNA technology. Okay, so first we will start from restriction enzymes. Since it is very important, we will start from that itself. Already in seminar 1 even I have said some basic concepts about this restriction enzymes. We will actually just elaborate that term. That's it. So what is a restriction enzyme? Restriction enzyme is actually a small classification in nucleases. Okay, it is a classification of nucleases. Okay, so it is a classification of nucleases. Well, what is a nucleases? Actually, any substance that is used to cut, okay, cut the genetic part, okay, the part which is in the nucleus, that genetic material DNA, okay, that we'll be calling it as nucleases. So a small, okay, a classification of this is what we'll be calling it as, okay, we'll be calling it as restriction enzyme. Restriction enzyme. Okay, so this restriction enzyme is actually of two different types. Okay, this restriction enzyme is actually of two different types. Okay, so what are those two different types? It is actually of endo. Okay, wait, I will write in a different color itself. So we shall write it in which color? Which color? Which color? Which color? Okay, so I shall write it in yellow color. Okay, so it is endonucleases. And then exonucleases. Okay, so endonucleases, and then actually restriction, before you will have to put the word called as restriction. Restriction. Okay, so restriction. Okay, so restriction enzyme is actually of two different types. One is restriction endonucleases, then another one is restriction exonucleases. Okay, endonucleases, then exonucleases. Now we will all have some doubts. Okay, not some doubts. Now you don't know what is the term. Okay, you don't know what is restriction endonucleases mean, and you don't know what is restriction exonucleases mean. Actually, these are very simple terms. Okay, see, first of all, what are their jobs? Their job is actually to cut a DNA. Isn't it? Okay, so restriction endo means inside. They will cut them inside of the DNA molecule. Well, wait, I will explain it still more clearly. We will first analyze them based on their nomenclature, okay, based on their name. So it will cut from the inside and this will cut from the outside. So that means what? This means, for an example, I will take a DNA itself. For an example, we will consider this one. Okay, perfect. Not that bad, right? It's somewhat okay. Or we will use technology. Turn, flip. Okay, so this is good. Here, if in case you are, okay, I will be also giving you all the examples of restriction endonucleases and then restriction exonucleases. Okay, now you are using restriction endonucleases. Then it will be cutting here, okay, here, center. Okay, here it will be cutting. Okay, so now you will be getting two different strands of DNA and lead. Then, okay, here after cutting this part, you can take it out. Then the desirable gene, they will put it in this particular part. Okay, then this particular desirable gene that you have put, right, that has to be sticking to this part and then to stick to this part. So that is why we will be using DNA ligases. Of course, we will be discussing about sticky ends, blunt ends, how to convert blunt end into sticky end, everything. Okay, and what does restriction endonucleases give, everything. First, we will just check about their general thing. Okay, so here in these two corners, actually, if in case this, see, now if you are using restriction exonucleases, then this enzyme will be cutting from the outside. Okay, from the outside it is actually used to cut. From the outside it is used to cut. So that is what we will be calling it as. Okay, that is what we will be calling it as. restriction exonucleases, which is known as molecular nibbles. Okay, you can also call them as molecular nibbles. All restriction enzymes are known as molecular scissors. Okay, well, we cannot, okay, this one also molecular scissor, this is also molecular scissor, this is also a type of molecular scissor. To be very specific, you can call them as molecular nibbles. Okay, NIBBELES, but that ERS, okay, that we don't have, so we will just take the out term. If in case they are asking in the examination, you will have to be sure what is molecular nibbles. What are molecular nibbles? Okay. Now, restriction. Okay, so we will start to discuss about them a little bit in depthly. See, restriction, we will have a plasmid. We will have a plasmid. This is a plasmid. Okay, so this plasmid is being divided into different regions. There will be resistant. Okay, here also there is a resistant area, and then there will be some other genes. There will be some other genes, ROP, ORI, then PAM, like that. All there will be different genes. Okay, different genes that will be coding for different things. But every gene is unnecessary for the cell. It's not like without this plasmid the cell cannot survive. The cell can survive without the plasmid. Okay, the cell can survive. Okay, so you take this. Okay, so for an example, a particular place, most likely it will be on plasmid only. So most likely, see, now you are putting endonucleases, okay, restriction endonucleases, then it will be acting here only. Okay, then it will be acting here only in this area, then this area. Okay, and here also it will not act everywhere. Like, you cannot say that this total area will be cut out. No. A very small segment of this area only will be taken out. Okay, very small segment of this area. Most likely that small segment of area will have 6 nucleotides here and then 6 nucleotides here. Okay, it will have 6 nucleotides here and then 6 nucleotides in this strand. Okay, so this particular strand, which is being recognized by the restriction enzyme, is there, right? That restriction enzyme recognizes this particular strand from this entire plasmid. So that is why this total region is called as recognition sequence. Okay, recognition sequence. Okay, so that is one thing. Okay, recognition sequence, and also it is having another name. It is also having, okay, another name, that is, yeah. See, this recognition sequence is actually, see, here also will be there, here also will be there, right? So here there will be some nucleotides. Okay, some nitrogen base pairs will be there. Here also some nitrogen base pairs will be there, and most likely there will be 6. Okay, there will be 6. For an example, best recognition sequence that I can give you is GATC, that they will be calling it as. Okay, we can call it as GATC, which is very good example. It is used in HIND2. No. HIND2 is different. Where is GATC? Where is GATC? One second, I'm just checking it out. GATTC. GATCC. GGAT. Okay. GAT. No, I can give you any example. So GATCC. Okay, wait a second. I will check. No, I don't have it here. Okay. So for an example, you just take this. Okay, you just take this, and then for this, okay, this is going to be. What is this? This is going to be. Okay. Actually, these two strands are having different names, right? I was the one who had taken seminar also on this topic. Everything I'm forgetting very nicely. Great. The only job that I'm doing is forgetting. So this is actually template strand and then coding strand. Yeah. So this is known as template strand. This is known as template strand. Okay, this is known as coding strand. Also, I've explained why did they get this name. Okay, so it's actually very simple. See this, for an example, you just conclude that this is their template strand. Okay, so this is the template strand, right? So you are having here G, okay, GGATTC. Okay, then here this will be having its own, okay, nitrogen base pairs, and that will be exactly the opposite of this, exact opposite in sense you just have to put it like, no, wait, then okay, it is going to be exact opposite of it. Yeah, here another C should come now. Now everything is correct. Okay, just put it exactly opposite, then C, like now you have written it from here to here, then now this time you will have to write it from here to here. So C, T, then here AA should come now. Arey Rama, even copying I'm not doing well. Very good. See, this one I have written here, but here I have written something else. Actually here it is very, very hot, so maybe that's the reason why I cannot concentrate. Okay, so C, G, A, T, C. Okay, exact opposite you have to write here. So C, T, A, G. Okay, you can see that it is exactly opposite. This template strand is actually opposite to here. Okay, to here. And when you are reading it, what is this? Okay, this will be, you can take it as 5 dash. You take it as 3. No, you take it as 3 5 dash. You take it as 3 dash. You take it as 5 dash. Okay, and then you take it as 3 dash. Okay, you can take it. Now just read it from 1 dash to another dash. So that means read it from 3 dash to 5 dash. Always read it like this. From 3 dash to 5 dash, and then it is GGATCC. Then from here also you have to read it from 3 dash to 5 dash. That is GGATCC. Exactly the same thing when you are reading it in the same sequence. So that is why, since if you read it from this side and if you read it from this side, it's the same thing. So we will be calling it as palindromic sequence. Okay, palindromic recognition sequence you can call it as. Okay. So, palindromic nucleotide sequence. Okay, so palindromic nucleotide. That is what the word palindromic means. Even if you are reading it from this side to this side, it means the same thing. If you are reading it from this side to this side, it means the same thing. Okay, best example: dad, mom. Okay, then Malayalam, which is given in our book, very famous example, Malayalam. Okay, dad, mom, Malayalam, everything is a palindromic sequence only. Okay, so that is why you can call this. Okay, so the sequence of nucleoside, okay, or the sequence of what shall we write here for restriction, for recognition sequence? They have even given the definition now. The specific base sequence is known as the specific base sequence that is being recognized by the specific base sequence that is being, okay, so that is being recognized by the, okay, so by the RE, okay, by the RE is actually known as your recognition sequence. Okay, so recognition sequence. Okay, so that is actually known as your recognition sequence. Okay, or I can also give you it as palindromic. Okay, I can also give the example as palindromic nucleotide sequence. Okay, so also known as palindromic sequence. And actually, if you just check here, where is it? Here it is there, right? I'm simply turning all the pages. Now I can turn it without any problem. Okay, so now we have like this, right? Actually here this will be cutting that when you put endonucleases, when you put endonucleases enzyme to here, then what will be happening is it will be cutting exactly here, okay, and then here. Okay, exactly, it will be cutting in these two places, and also mostly, as I said, that it will be having 6 nucleosides only, okay, in template strand and as well as in the coding strand. Okay, so here, now you are having like this. Okay, so how will it cut then? It will cut like this. Okay, so this is one way. Then here, this is also another way. Okay, so this is the way how they will be cutting. So you can just separate. See, actually, what did I say? That particular part will be taken out. I said, right? Actually, that's not the way. Sorry, I said it wrongly. To understand in that particular point, I said it like this. Actually, see, there will be crack. There will be a crack. By that crack, you can actually, in this crack only, where now it will be like this. Okay, it will be G, C, C, T, A, G. Then here it will be G. Here it will be G, A, T, C, C. See, now you can combine it. You can separate it. You can combine them. You can separate them. Okay, and you can actually see these ends are known as sticky ends. Sticky ends. Okay, this is also known as sticky end. Okay, and also there is a big misnomer. What is a misnomer? A wrong knowledge, we can say. That is what misnomer will be calling. Okay, so there is actually a misnomer in here that is where are you? Here you are. That is restriction endonucleases always give you only sticky ends. No, that's not correct. Restriction endonucleases gives both, okay, both sticky ends and as well as the blunt ends. And here you cannot say any sticky ends and any blunt end, but because see, you are actually having it chopped from the outside. Okay, you are actually having it chopped from the outside. Now you will be having two different parts of DNA when you are using endonucleases. Okay, only one segment. This one segment you can take out. Okay, from the desirable gene you can take one segment, but when you are using like this, here you cannot do like that. Here you will be having this as your segment and this as your segment. But in this case, in endonucleases case, you will be having only one segment. So that is why, okay, this will not produce. We cannot say that it will be producing you a blunt end or it will be producing you sticky ends. But here, this restriction endonucleases can produce both. Okay, it can produce both sticky ends, okay, and the blunt ends. Okay, there is no doubt in that. It produces both the sticky ends and as well as blunt ends. Okay, so now I have said you what is sticky end. What is sticky end? Okay, so this area, this area is what we'll be calling it as sticky end. Okay, so it'll be like when you're actually having it over one, when you're overlapping, they have to again add. Well, here you will not just like that overlap, you will actually use here the desirable gene. Okay, that desirable gene will be used and it will be filled over here. Okay, and to stick, okay, this desirable gene along with this particular DNA, the DNA of your plasmid, you will be using DNA ligases. Okay, ligases, which is used to stick this one and then this one. Okay, so then what about blunt ends? Blunt end is the part where like this it will be. Okay, here you can have I will be giving you one example, that is, okay, so G, T, T, and then here you'll be having A, C, Okay, here you'll be having G, T, T, A, C. Okay, so like this you will be having C. Here, there, as I said, that restriction endonucleases itself, restriction endo, this enzyme is there, right? This enzyme, restriction enzyme, is actually of two different types: restriction endonucleases and restriction exonucleases. Here, this restriction endonucleases is having some varieties. Okay, so those varieties have their own varieties of palindromic nucleotide sequence. Okay, so some varieties, and also same. Okay, this restriction exonucleases will be also having their own varieties of palindromic nucleotide side. It is supposed to be side now. No, tide will be correct. Okay, so nucleotide sequence. But we can say that exonucleases mostly, okay, mostly when you are taking here, when you are taking it like this, okay, when you are taking this as the fragment, okay, here you are having cut, here you are having cut, in that time you can say that in some cases restriction exonucleases produces blunt ends. Okay, that actually also produces blunt ends. Now ligases, this, start, everything. Okay, so now you have got this blunt end, and blunt end you will use nowhere in the entire biotechnology field, because this DNA ligases will not work on this blunt end. That is the reason. So to convert this blunt end into sticky end, into sticky end, what generally we will be adding is, we will be adding alkaline phosphatase. Okay, alkaline phosphatase enzyme. We will be adding alkaline phosphatase enzyme. So what will this enzyme do? This enzyme will not do anything that is great. Okay, actually, like you cannot, this is not that much complex thing, it is very simple thing. Actually, this enzyme will add here adenine, and here also adenine. So because of this addition of adenine here and here, now this has become as sticky ends. Now you can add any desirable gene to this particular part. Okay, and and here exactly this is the site where this restriction enzyme acts, and here also this is the site where restriction enzyme acts, right? So that is why this site is actually known as restriction site. Okay, so restriction site is a particular point in a recognition sequence where restriction enzymes act on this particular point to separate the DNA molecule. We can say it like that also, right? So restriction site, where are you? Okay, so I think I have covered as much as possible. Okay, everything that is possible. Okay, so these are very, very important. Now I'm going to give it in a good notes. You can see that how horrible it is. Okay, here it is really, really horrible, right? We will be writing it over here like a summary. Okay, so the specific site, which color? This color is okay now. The specific where RE restriction enzyme acts is known as okay, restriction site. Okay, is known as your restriction site. Okay, so then you will be having here, now when you use restriction endonucleases, endo, then exo. Okay, exo. Here you will be having both, okay, both blunt ends, then also sticky ends. Okay, so to convert this blunt ends into this sticky ends, okay, into this sticky ends, what will we be using? We will be using alkaline phosphatase. Okay, so alkaline phosphatase. Okay, so alkaline phosphatase is the enzyme that is being used. Okay, alkaline phosphatase that is being used. And in exonucleases, if they are asking you in multiple choice questions, then you can write it as blunt end itself. Okay, you can write it as blunt end itself. Okay, blunt end is the thing. Now then, what else have I left? Still there are examples. Still there are some points that I know. Palindromic sequence I've written, that I've written. And also this I have said in the previous seminar itself, but here now I have not yet said, that is, see, to get desirable gene, okay, to get desirable gene, see here, this is what your desirable gene, right? So to get this desirable gene, what you have to do, you will have to actually use restriction enzyme, okay, that too endonucleases, restriction enzyme only you will have to use. And that same restriction, that same endonucleases restriction enzyme only you will have to use also here to have this crack. You can see that there is a small crack, okay, there is a small crack here. How beautifully have they made this image, even it depicts the crack. Okay, so here this enzyme, the enzyme which I have used here, that same only you will have to use here also. Okay, if you are using some other different thing, then DNA ligases will not work. Then DNA ligases will not work. Okay, it will not work. Okay, so that's one point. Now we will come to something, see, some history points, that is, in the year of 1963, in the year of 1963, only. So in the year of 1963 only, they have found two enzymes that are responsible for the restricting the growth of bacteriophage. See, they have found two different enzymes. Okay, in the year of 1963. Okay, so scientists have found two different enzymes. Amongst them, one is restriction enzyme. So amongst them, one is restriction enzyme. Then what about the another one? That is actually methylase. Okay, methylase. Actually, in methylase, even you are having two different categorization that we don't have, but the before name will be having DMC, sorry, DMS. Then you will be having here DAS. Okay, DAS. Just give me 1 second. I'll just check it whether it is correct or not. Okay, so as I have expected, this is wrong. It is supposed to be DAM and then DCM. It is supposed to be DAM and then DCN. So what is this methylase? Okay, see this one. Okay, this one still. Okay, first I will complete what is this restriction enzyme's role. Then we will be coming to methylase. Okay, see this restriction enzyme also. There is something that we will have to discuss later also. But now we will be just. I will just go then. Go on, and later I will be saying some other points. So first type of nuclease, that is first restriction enzyme that they have found, is actually none other than our famous HIND2. Is our HIND2. So as I said, right, so there are some specific names for restriction enzymes that more than this HIND2, lots of things are there. I will be giving you around 6. We will be reading all of that. Okay, so HIND2 is there. So this HIND2 is a type of restriction endonucleases. Okay, so restriction endonucleases, that is there. Then other than that, they have found, okay, they have found 900, not 1 or 2, they have found 900, okay, 900 restriction enzymes from just 230 strains. What does the word strain means? Many people confuse over here, so that is why I'm stressing here. So strains, what does the word strain mean? Strain means, you can say it as varieties of bacteria, okay, to understand it in terms of simple terms, okay, it is varieties of bacteria. That means from 230 strains they have found 900 restriction enzymes, and each restriction enzyme is having its own, okay, its own palindromic sequence. Okay, so now we will be going to here. This is our very important slide. He is the hero of this lesson, E. coli. Okay, so E. coli. See here, actually when a bacteriophage comes, okay, so this is our big boss bacteriophage. Okay, so here, okay, this will be having its own genetic material. I hope you all know that. Okay, so this genetic material will leave here. Okay, actually this will not be this much small, it will be bigger only, but there is no space, and I'm a very good drawer here, so I can draw like this only. So this will leave its genetic material over here, so that this genetic material can come here and it can come by. Note, it will not go here. Wait a second, I hope I am correct, I'm not wrong. Yeah, I think. You might be thinking, I will check that also. Okay, so what I've said is correct only. Actually, that is how even it will be going. Okay, I have no basic common sense. Come on. Okay, see, when it goes here, it cannot divide. Okay, just because of increase in number of plasmids, there is no such need as, if there is increase in plasmids number, then that doesn't mean that there should be increase in the cell number also. Okay, but here the bacteriophage wants to duplicate, or it wants to increase its size. Size of the population. Okay, so for that, here only it has to go. So whenever there is increase, or whenever, we all have studied cell division, so whenever cell division happens, then automatically this one's number also increases, because this, its DNA is what here. Okay, so this bacteriophage leaves this DNA, but this cell is actually very brilliant. This E. coli is actually very brilliant. We will be studying about recombinants, or then transformants, then non-recombinants, okay, non-transformants, everything we'll be studying. Okay, I'll be saying that, don't worry. So here, this DNA is over here. Okay, so if it comes, then automatically this E. coli will not be under its control. It will be in the control of this particular gene. So to protect itself, this E. coli has developed two different enzymes. Okay, that is going to be our famous RE, restriction enzymes. Okay, actually, there is also in cells, there are organelles, right? Okay, I've completely forgotten everything. There is nucleus, there is mitochondria, then surrounding to this cell here, which creates this cell membrane, is actually RER and then SER. But I've forgotten the name. I have that short form alone in my brain. Endoplasmic reticulum, rough endoplasmic reticulum, and then smooth endoplasmic reticulum. So do not get confused with that. Okay, I was thinking of that only. Okay, then there is actually also another enzyme, okay, that is actually responsible for saving itself from this RE. This RE is produced by this E. coli only, and this, here this E. coli will produce from this plasmid. Okay, I think from this plasmid only, there is no data about it. Okay, so with me, okay, not in our there is no data in our NCERT book, I think. Okay, till what I've read. If in case there is data in the future lectures, I will be giving you. So this plasmid will be actually secreting RE, restriction enzyme. That restriction enzyme actually goes and it cuts the part of this DNA. And when more than one part is being cut in this DNA, then it will be very hard to ligate it. To ligate it, it will be very hard, and this DNA should be ligated. If it is not being ligated, then this DNA's function will not work. DNA's function will not work. The DNA molecules has to be together with each other. If you just separate the DNA molecules, then their function will not work. Okay, so this RE will be destroying itself, and actually this RE has no feelings like that at all. Okay, so this also tries, and it also tries to destroy destroy this linear DNA also. So to save this DNA from its own RE, what the cell has done is it has started to produce a particular enzyme called as methylase. Okay, methylase. The function of methylase is actually to add methane. Okay, methane to the circular DNA. Sorry, to the linear DNA. To the linear DNA. Okay, so here it will be adding its linear DNA. Okay, so here, as I said, that there are two different things, that is DAM, okay, DAM, and then DCM, where here this, okay, DAM methylase, okay, DAM methylase will be adding methane to its, okay, will be adding methane to the adenine, okay, to the adenine. Now this DCM methylase will be adding methane to its cytosine, See, C and then A. This D and M are constants. This A, the center point only, will be varying. If here A is there now, then it will be adding to adenine. If C is there now, then it will be adding to cytosine. Okay, so this is what its total thing is. Within that, I've got aged. Okay, so this is what we will have to discuss now. I hope this handwriting is pretty good. Okay, so we will talk about types of RE. Okay, so types of restriction enzymes. So there are totally two different types, endonucleases and then exonucleases. That is what we have seen. Okay, I'm going to give you the examples. Okay, so endonucleases and the exonucleases. Okay, so coming to its examples. Okay, so first of all, I will be giving you examples for here. Okay, so we have a few. Also, see, very common, and also we have nomenclature. Yeah, okay, thank God I didn't forget it. Okay, so after nomenclature, I have to explain now two different images. Then also we have another thing. Then only we will start cloning vectors in our next video. Okay, already I think it has been 40 minutes. It has been 42 minutes and 38 seconds. Okay, so we will start here itself. Okay, so endonucleases, eco R1. Eco R, not just one, double one is there. But eco R1 is a very famous example, by the way. Okay, so this eco R1 is a very famous example that you will see. Okay, then after that, eco double one only I have here. Okay, so then after that we have Hind 2. Okay, Hind 2 I will be giving you later. Then we have Hind 3. Okay, we have Hind 3. Okay, then we have BAM HI. Then also we have H, like ha. Okay, so Ha1. Then we have LU. Okay, LU1. Then we have Hind 2, which is what had been discovered in the year of 1963. In the year of 1963 they have discovered this only. Then over here we are having Ha1. Then we are having Ha1. Sorry, Ha3. Okay, this is not Ha, this is Ha. Okay, Ha3, then Pa3, then Ha1, then BAM. Okay, something like that. Okay, so we are having all these, right? See, as I already have said to you, that endonucleases actually can produce both blunt ends and then sticky ends. Then this exonucleases can produce mostly, okay, according to our MCQ knowledge, only blunt end. Okay, so here I have also categorized them based on this. Okay, that is, here you are having which color? This color. Okay, here you are having sticky ends, okay, then blunt ends, okay. And if in case your teacher has told you that endonucleases can provide only sticky ends and exonucleases can provide only blunt end, then I'm so sorry, that's wrong, okay. And if he doesn't accept it, then go along with him even, okay. Like in the exam paper you will have to write the same thing, because teachers are those people whom we have to respect from our heart, okay, if that teacher is acting like a teacher, okay. So we are having eco double R. So best example that we can give is NCC AGGN. Okay, first I will write, since it is all palindromic sequences, so we will first write only one sequence, okay. Then H333, so A G C T T. Wait, I will write A G C T T. Then for BAM we will have G A T C C. See, little bit of difference when we both are having. Then for here, okay, so there is no space. It's okay. Okay, so here you have G. Okay, so ha G C G C. So it is having, see, one of the cool factors that ha is having only 3. Okay, so then after this we also have here alu is actually AGCT here also AGCT. Then our hint 2 is having but 6, and hint 2 is very unique from everything. Okay, hint 2 is actually, and also I can give you also proof that this endonucleases is able to give both sticky end and blunt end. I can give it very easily. I will give you now. Okay, don't worry. See, hint 2 is actually G T pi, then pu, okay, A C. Okay, A C. Then here, okay, that's it. Okay, so what is this pi and what is this pu? Pi is pyrimidine, pu is purine. Okay, pyrimidine, purine, we all have studied that, right? Purine is where we will be having A and G. Okay, pyrimidine is where we will be having rest of the 3. Okay, our heroes of our biomolecular basis of biology. Okay, so then what about their remaining things? Okay, here you will be having N. Sorry, wait. Oh God, how is this possible? 3 N now. Eco R2N. What is N? Cytosine, adenine, guanine, thiamine. Is there something like N? Oh God, have I forgotten it? Have we read it in our biomolecular? Okay, so I did check it. But I could not get any answers. So, but here for eco R1 I got. Okay, for eco R2 we have like that. We have also, I have got also for eco R1. Okay, so we will start from here itself. Eco R1. Eco R1 is producing sticky ends mainly. Okay, so for here, how will you write it? You can write it as C T T A G. Okay, so here by you will be having, it will be cutting over here. Okay, in this place exactly. Okay, like this. you will be having it like this. Okay, so then how about here? Here you will be having N, here also you will be having N only. Here you will be having G, G, A, C, C. Okay, and hereby it will be acting on here. Okay, here and then here. That is what I have heard. Okay, not heard, that is what I have written here. So, but I'm not sure what is N. Maybe N can be the rest of the DNA. It can be the rest of the DNA. It can be, or it can be something else. But what can be N? If it is pi now, we can say it is pyridine. If it is pu now, we can say it is pyridine. But pyrimidine and purine, not pyridine. See, okay. But what about that? I cannot say anything for that. N. I really don't think I've written anything like N. Thymine, okay. Adenine, okay. Guanine, okay. Cytosine, okay. Even another one is also, wait. Another one will not come here because that is only for exclusively for RNA. Then over here, what can be? Okay, so here we have T, C, G, A, A. Okay, so hereby like this only it will be C. Here it will be actually acting only in the corners, like before the corner, and you need not even remember everything. Okay, like I don't think other than for what they have given in our NCERT, okay, which is given GAA-TTC. Where is our GAA-TTC? Oh, so this is what's given, okay, GAA-TTC. So this is what they have given for in our NCERT. So this is what very important. I think so all of them, just remember that EcoR1, which produces sticky ends, which is a type of endonucleases, which is a part of restriction enzyme, which is a part of nucleases. Okay, that will be having this as its sequence. Okay, then what about here? C, C, T, A, G, G. Okay, here. Okay, then here also you will be having it like this. Great, I cannot even know this. Okay, so this is going to be G. Okay, this is going to be C. This is going to be G. This is going to be C. See, it is like alternative. Okay, it is alternative base pair. Okay, and here it will be acting here, and it will be, oh god, okay, it will be acting like this. I hope you all can see it. Okay, if you all cannot, then I'll erase this. Okay, so you will be having like this. Okay, then papa, no, you are having an ALU, so it is T, C, G, A. Okay, like this. Then what about here? See, here it can be any pyridine or pyrimidine, or it can be any purine. Okay, but it cannot be the thing that comes for RNA, and I forgot what is that particular thing. How can I? So it can be A, G here. It can be here C and T. Okay, it can be. Okay, so here C, A, pu, pi. Okay, pi, T, T, G. Okay, so here it is acting like oh yeah. Here how come? Here actually see, this is blunt end na, so that is why see blunt end na. So here will be center. Here also it will be acting on center only. Like this. It will be acting like And also Hind 2 is an example that is given in our book itself, and they have exclusively said that Hind 2 is actually a part of endonucleases. Okay, this is a part of endonucleases. See, okay, endonucleases. And here what have I written? Here what have I written? Hind 2 is actually giving us blunt ends. So that means what? Also endonucleases. Actually, by looking at this Hind 2 only, I got doubt. Even I was thinking like that only. Okay, but then I asked my teacher, then she said that yes, endonucleases can provide both sticky ends and it can provide also blunt ends. Okay, it can have both. Okay, she said, then only I got okay. So this is the case. Okay, now we have also PA. So PA is GTTAAC, 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 oh, GTTAAC. Here C T T A A G. Then he is G G C C C. G G C C C C G G. Okay. By the way, everything over here is actually phi dash. This side, okay, this side. In the template strand is phi dash. This side is 3 dash. This side is 3 dash. This side is phi dash. Everything, there is no exception in that. Okay, but here I've taken it wrongly. Here I've taken it. Here I was where was that? Here na. Here when I was explaining, here I've taken it wrongly. Okay. So here, what will be happening? Here it is actually producing only blunt ends. Here you will be having only blunt ends. Okay, so exactly here it will be acting. Okay, here it will be acting, and here it will be acting. Then for here it will be acting exactly TA. Okay, T. Okay, and also one more thing I have to say. See, the mechanism of this endonuclease is very simple. It developed, okay, it has the power, okay, it has the energy to break only one hydrogen bond. That bond is very unique. For each and every restriction enzyme, its specific restriction site is different. Okay, so that site will be for one restriction. Restriction enzyme, there will be only one restriction site. Okay, so that restriction site is what here GA. So it has, see, EcoR1 has only the power to break the bond between GA or AG. Okay, so here that is what is happening. Here also same. See, AG or GA both are same. Okay, like this, then like this. In the hydrogen bond they are same. Okay, so here also see, here it is NC, here also it is NC, here also it is AA, here also it is AA, here also it is GG, here also it is GG, here it is GC, here it is GC. But we have to read it. See, if you are reading it here like this, then here you have to read it like this. That you have to note. Okay, so everything is same, aren't they? Everything are same only. Okay. So next is one more thing, that is nomenclature. Okay. So very important thing is nomenclature of RE, and yeah, as soon as possible I will be also putting nomenclature for inorganic chemistry, nomenclature in organic chemistry, which is very important by the way. Okay, nomenclature of RE. So here actually it is very simple. You just take the case of EcoR1. Okay, you just take the case of EcoR1, which is a very famous example. That is what we have seen. EcoR1. Okay, so here see we have so the first, okay, the first letter, okay, the first letter, this is eco is there, right? This is actually none other than its genus. Okay, Escherichia. Okay, Escherichia. Then the second one is none other than our species. Now you all might have a doubt. What are you doing, Kannan? Whether restriction enzymes are having genus and species? What? Okay, you say that it is just an enzyme, then how can they have genus and species? Actually, they do have genus and species, because you are not writing the genus and species of restriction enzymes, because that is a substance, and substance don't have genus and species. It's nonsense. Okay, you are writing the genus and species from where you have extracted RE. RE is produced in E. coli. Okay, RE is produced in E. coli. So that is why. Okay, RE is produced in E. coli. Then what about R? Okay, yeah, what about R? Okay, your R and then your 1. R is your strain, and as I said, okay, as I have already told you, that strain is a type of bacteria. See, like a dog is having a different genus, and you cannot say that Doberman is a species of dog. It is a subspecies, you can say, or it is a part of dog, we can say. Even in humans, we had some categories. Okay, even in Homo sapiens, I think even now one more. No, there is no such thing. Okay, in dog, you cannot say that Doberman is a species. Okay, it is a subspecies. Okay, like that. Even bacteria are having their own subspecies, like Doberman, like Pug, like Pomeranian, like Chimpanzee, like Rajapalian. Okay, then here 1. So 1 is where you are having, okay, order of isolation. Yeah. Order of isolation. What is order of isolation? See, actually in olden days, scientists weren't as classy as we are at present. So what they did is they will be extracting lots of substance from cells. Okay, because that was the time in the 20th century was the time where the cell evolution, like cell evolution in cells, the cell study, chromosomal study, then this cytoplasm, everything. Okay, everything. Cytoplasm we have discovered a little bit before even, but lots of parts were discovered. So each and every scientist were very indulgent in discovering each and every part. So what they did is, in order to not to get any confusion, what they did is they extracted that particular part and they have kept it in a particular capsule, and they have given that capsule order, name order. Okay, 1, 2, 3, 4, 5, 6, like that they are given, and from which capsule is this one. Okay, that is what this is. So we also have Eco R2, so that means it is in the second capsule, and Eco R1 is actually first capsule. So these two had been taken for study, and they have checked that these two have some difference. So they have kept it as Eco R1 and they have kept it as Eco R2. That's it. Okay, so this is what their nomenclature is. Isn't this very simple? Okay, so I hope I have explained already this. Okay, I've explained this many times. Okay, so but again I'll be explaining it. This is our desirable DNA, this is our plasmid. Okay, in this desirable, in this chromosome, this is our chromosome and this is our plasmid. In this chromosome and then in this plasmid you will be putting RE, which is your restriction enzyme. Okay, and that is going to be endonucleases only. Okay, so that restriction enzyme will be acting on this plasmid, and this plasmid you can see that there is a small crack over here. Okay, and here you will be able to separate your desirable gene from this eukaryotic chromosome. Okay, and that eukaryotic chromosome you are going to insert it into this plasmid, then you will be having your rDNA. Okay, so this is your rDNA. Okay, so this rDNA, by some methods you will be putting it inside. Okay, you will be putting that inside this cell. Okay, it can be any cell. Okay, see, transformation is the procedure through which a piece of DNA is introduced into host bacterium. See, so this is being introduced here, right? Okay, well, bacteriophage does this transformation only. Okay, here bacteriophage is there. Okay, so what does it do? It leaves its DNA inside. Okay, so while leaving its DNA inside, this process, that process is what called as that process is what called as transformation. Okay, so now what happens is this cell will start to divide, and while dividing also this plasmid will divide, and also this plasmid is having its own gene. Okay, but generally this will not go under this gene. Okay, this bacteriophage gene will not go here, but instead of that will be going here. Okay, but we can do it here also. Okay, and then here it will be, and then these cells will start to produce the required product, or these cells will have this plasmid, and this plasmid, see, plasmids are, as I said, they are protective barriers. They secrete some substances to protect the cell, or they secrete the thing that is in the gene, okay, by using the resources in the cell. Okay, so here, if you have inserted here insulin gene, then here you will be having insulin. Okay, in cell engineering also we will be studying about X-gal, we will be studying lots of things are there, we will be studying it later. Okay, so this is your procedure. Then also we will be talking about this. See, molecular scissors. We have got here actual scissors itself. Okay, so what will be happening? You will be having a DNA. You will be having here also DNA, and here we have got the elaborated form. And also, each color has its own meaning. Okay, I suppose each color has its own meaning. Okay, so but that at all we don't have. Okay, so here what will be happening is this RE, okay, it can be anything. So maybe if you have got it, it depends upon the sequence only. We will be just remembering GAATTC. So GAATTC. Here also you will be having, see, here it is what target site. Here this is what target site. Here also you are having GAATTC. GAATTC. Here also same thing. Okay, sorry. Here it should be exactly opposite. CTTAAG. Here also like that only will be having. Okay, so these two will be. Okay, so these two because of this eco R1. Okay, so this particular part will be removed. Okay, this particular part will be removed, and here this particular part will be removed. Then you will be having like this, and this area is what called as sticky end. Just like in enzymes we have studied, right? Okay, so reactive site. Okay, everything we have studied in enzymes in biomolecules. Like that only this is also. Now by using DNA ligases. Okay, by using DNA ligases. Okay, this you can combine with each other. Okay, so also this one you can use, see, this one you can say it as target site. So that means this is what your desirable gene is, and this is what your eukaryotic chromosome is. Okay, so you have got your rDNA. Okay, you have got your rDNA. This also we have completed. Now we are entering into our one of the most finest topic, which is our gel electrophoresis. Okay, gel electrophoresis. By the way, before entering here, no, that we have in plasmids. In plasmids, we'll be discussing types of plasmids also. Okay, so this is actually a typical process by where you are using this in fingerprinting. Okay, this is a process. Actually, this is everything I have studied in biomolecular basis. Sorry, there in that chapter even biomolecules, even not biomolecules. Okay, molecular basis of biology. Molecular basis of biology. Over there even we have studied. Okay, so the same diagram I think we have there, but with I think the same diagram only. Okay, so here this time, not the same all the time. This is your anode. Okay, this is your anode. Okay, and then this is your cathode. Okay, so what are these? These are DNAs, aren't they? Okay, so you have put this DNA over here. You have put this DNA over here. Here you are having agarose. Okay, here you are having agarose. Agarose, na? Yeah, agarose only. Everyone would have eaten jelly, or at least they would have heard about jelly. My favorite thing is that jelly only. Okay, it is really very good, and we can all make it in our home even by using this agarose. Okay, this is actually a substance that is being extracted from the seaweed. Okay, so seaweed. It is extracted from the seaweed. Seaweed in Tamil we will be saying it as kadal pasi, I think. Okay, so it is being extracted from that only. It will be transparent. Okay, you can boil it. You can put it, you can boil it, and you can add some sugar with water. Okay, then you can put it in the fridge. Then it will be solidifying. Then after that, you will be getting a jelly. Okay, so this is what it is, and also along with that, yeah, this agarose is what actually is there. Okay, and then here you are having this DNA. Now, as the electricity started to pass, since you can see that here it is what? Here it is negative charge. And what are these DNAs? These DNAs are also negatively charged because of the proteins that they have. Okay, negatively charged DNA is also negatively charged. Okay, so now this negatively charged and this negatively charged will do what? They will actually repel each other. There will be repulsion. So because of repulsion, the least weighted amongst them, you can see that the small fragments, the smallest fragments goes the farthest distance, the furthest distance. Okay, it goes the furthest distance. And the highly weighted goes the least distance. Because it is highly weighted, okay, this repulsion force is not required. It is not the optimum amount, okay, to push it till here, okay, so till this much distance is there. Okay, so actually this is actually, see, wait, one second, I'm seeing lots of actually so by gel electrolysis, okay, by gel electrophoresis, by this method there is an effect, okay, by that effect only these all are moving, these all are being separated, okay, these all are being separated by that particular effect, all these DNA fragments are being separated from each other by that effect. That effect is what called as sieving effect, okay, this is also a key point, this is also a key point. Okay, it is sieving effect. By the sieving effect only movement happens. Movement happens. Okay, so movement happens. So here it goes. Then also see one thing, this DNA is not visible, it is not at all visible. So to make it visible, okay, so to make it visible, which color, I will use one of the darkest colors, that is this. Okay, so to make it visible, what we all will be doing is, actually we will be adding a particular substance called as ethidium bromide, okay, ethidium bromide, okay. Then after this also we will add UV rays, which we will be studying in our physics, okay, and also we all know what is UV rays. We will be studying in physics, in electromagnetic waves we will be studying, okay. So UV, so when you are adding this, automatically this DNA will be visible, okay. When you add these two, this will become visible in bright orange color, okay, so in bright orange color, bright orange color. Okay, so I hope I have said each and every point in our NCERT book as much as possible. So that's it for today's video. We'll be meeting in another amazing video. Till then, bye bye.